A biological membrane is made of three components, namely the phospholipids. These are these ones over here. It's a phospholipid. Proteins, these here. And cholesterol, and these are the yellow structures over here. And of course phospholipid. So if you mix membrane proteins, cholesterol and phospholipids, if you mix them together in water and you shake them, then they will go automatically into a phospholipid bilayer membrane, and the proteins will be embedded in the bilayer and the cholesterol as well. The question is now, what holds it in place? The membrane arranges automatically because it's the most stable configuration, and nothing holds it in place other than the fact that the phospholipid heads are water soluble and the cholesterol is not water soluble, it is hydrophobic. So it will go into that arrangement on its own. Important is the following, that the protein itself, if it is to stay in the phospholipid bilayer like this, must also have hydrophilic and hydrophobic parts. So this part of the protein over here, the outer part over here, is also towards the water side and must be hydrophilic, just like the heads. And this part over here of the protein, this outer part over here as well, is also hydrophilic, and the hydrophobic part in here, let's put it this way, rather on the side, the one which is turned towards the hydrophobic tails, this must be hydrophobic of the protein, and this part over here as well, because it is next to the hydrophobic tails of the phospholipids. So that is basically one thing. The question is now, what is the function of the cholesterol? And the function of the cholesterol is twofold. First of all, the cholesterol makes the membrane less fluid. Now, what does this mean? The phospholipids are not bonded together, but are able to move around freely inside the membrane. The proteins as well. And they're held in place by the fact that the heads are hydrophilic and the tails are hydrophobic. But on their own, they are able to move around the membrane. And cholesterol, the presence of cholesterol, makes the central part of the membrane more hydrophobic, and this also reduces the fluidity of the membrane. It makes the membrane less flexible. Maybe if I use this term to explain this, it makes it less flexible. The second role that cholesterol has is that because it makes the inside more hydrophobic, hydrophilic substances, just like salt, do not want to diffuse through the bilayer membrane. So let's say that this is sodium Na+, which is very polar. It has a positive charge, very hydrophilic. It's dissolved in water and it has no problem over here, staying over here. But it does not want to go through the phospholipid bilayer membrane, because it then would have to pass the hydrophobic central part, and it's not going to do that. So if I draw an arrow like this, it's not going to happen. Okay, I'm going to cross this out here. Okay, the sodium is not going to diffuse through, also because the cholesterol over here makes it even more hydrophobic. So how can the sodium over here, the salt, how can it now reach the other side of the cell membrane? Well, that's why we have these channel proteins, and what you see in the middle over here is a channel, and I'm going to erase now these parts over here again to make it a little simpler. And what we have is the following. This is a channel protein, and the channel protein now allows the diffusion of the sodium to the other side, because the inside over here of the channel protein, the inside lining, so to say, is hydrophilic. Remember that what I just told you before, that the outside over here is hydrophobic because it's next to the hydrophobic tails. Yeah, but the inside is hydrophilic. So the sodium, I'm going to draw this again over here, Na+, has no problem diffusing through the membrane to the other side because the protein, the channel protein, allows it to do so. So that is basically the significance of a membrane, and also how it's able to control the passing of substances, because certain substances can only pass through specific channel proteins, and if these channel proteins are not there in the membrane, the substance is not able to go across. So it's locked out, so to say. Okay, so that is basically the overview of the membrane. And when you draw the membrane, and just the last little tip, some students draw the phospholipids like this. They draw a phospholipid head, then they draw the tail, and they draw the tail, right? That's how you draw a phospholipid bilayer membrane. Horrible, horrible, horrible. Don't do that. But I get drawings like this. Multiple problems. First of all, the phospholipids don't touch each other. Over here, the phospholipid is actually not one molecule, but the two fatty acid tails, these are fatty acid tails, are not connected to the head. So it's a really bad drawing. So be careful when you draw the phospholipids, and do not draw it like this. Okay, so basically this was it here.